Hello, my name is Savannah Orton, and today I will be discussing the comparative analysis on extreme codon usage biases in tRNA genes found in actinobacteriophages. Bacteriophages are viruses that infect bacteria hosts in order to replicate. Phages must rely on the reproductive machinery of its host to reproduce virons and proteins, and will likely have trouble replicating in a host whose codon usage bias is very different from its own. The presence of various tRNA encoding genes may confer a selective advantage to a phage by allowing it to infect and replicate in a wider range of hosts, thereby increasing their ability to survive in diverse environments. In this study, tRNA encoding genes from a variety of phage clusters are analyzed to widen the current knowledge of extreme codon usage biases in phage-encoded tRNA genes and host codon usage biases. Furthermore, tRNA anticodons were compared to the codon usage of the phages and potential environmental bacterial hosts. To conduct this research, I utilized DNA Master and a program called HiveCuts to generate the codon bias tables for the phages and bacterial species respectively. In addition, tRNA genes were detected by sequence analysis with tRNA scan SE and Aragorn. Here is a table that lists every phage I analyzed throughout this research. Amigo, Gorgeous, and Convoy were all isolated in an Arthrobacter species, while Eurytus, Emmanuel 3, and Newt were isolated in Streptomyces griseus. This is a visual representation of the programs I used to generate codon usage tables. On this screen, I present codon usage tables for each of the bacteriophages analyzed. Several of the phages demonstrated distinct codon biases with compensating tRNA genes. In the generated tables, green, yellow, and blue colors were used to highlight extreme, moderate, and low codon biases respectively. Phages Emmanuel 3 and Newt possess a greater number of tRNA encoding genes and show multiple extreme codon biases. For example, Emmanuel 3 shows an extreme codon bias for AAG lysine codeine at 99.9% usage. Likewise, Emmanuel 3 shows an extreme codon bias for 10 other tRNA encoding genes. These are the percent codon usage tables I generated for the bacterial species using HiveCuts. These are two more codon usage tables generated by a previous student from our lab. For an actinobacteriophage to survive in a host bacterium, it is expected to see a similar codon usage between the phage and the host. However, codons for which Arthrobacter species or Streptomyces griseus has reduced codon usage are frequently compensated by tRNA genes encoded within the phage genome. This observation can be observed in Newt, which has an extreme codon bias for CCA proline codon at 35.5% compared to Streptomyces' usage of 1.8%. Phage Newt encodes one tRNA gene that has an extreme codon bias, 9 that have moderate bias, and 1 that has low bias when compared to Streptomyces species. Amigo and Gorgeous did not encode any tRNA genes that demonstrated an extreme bias when compared to Arthrobacter species. However, extreme codon bias was observed when compared to host Gordonia terrae and Mycobacterium foliorum. Finally, here is a table that depicts the host relative codon bias strength in phages with tRNA encoding genes. Phages need to utilize their bacterial host genetic machinery in order for a phage to survive and replicate. Phages often take advantage of the host codon usage to regulate the expression of proteins. If the host does not display a sufficient level of charged tRNA genes to translate the phage's gene products, the expressed tRNA genes within the phage's genome can compensate for the disparity between the codon usage in the host and the codon usage in the phage. The presence of a relatively high number of tRNA genes in phage Emmanuel 3 and Newt demonstrate moderate to extreme codon bias between the phage and Streptomyces griseus supports this hypothesis. Seemingly, the tRNAs in the phage genomes compensate for a discrepancy in codon usage in the host bacteria. Phages Amigo and Gorgeous did not demonstrate any extreme codon bias when compared to their isolation host, but displayed extreme codon usage bias for 2 tRNA genes when compared to potential bacterial hosts, Gordonia and Mycobacterium. This observation may explain the phage's ability to replicate in multiple hosts despite their isolation host. In the future, phages Amigo and Gorgeous could be investigated to see if these phages would show an extreme codon bias for the phage's tRNA encoding genes in a variety of hosts. Conducting this research may provide more information as to how wide the actinobacteria phage host range is. Finally, I would like to thank the Howard Hughes Medical Institute for acceptance of LeTourneau University into the C-Phages program, the Pittsburgh Bacteriophage Institute at the University of Pittsburgh for sequencing the phage genomes, LeTourneau University's Biology Department, and my two professors, Dr. Ballerine and Dr. Frederick, for helping me throughout this research. Thank you.